This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we're diving into one of evolution's most intriguing puzzles. Why does sex exist? It may seem obvious, but reproducing sexually is costly and inefficient compared to simply cloning yourself. Yet most complex organisms do it anyway. The answer might lie in an evolutionary arms race. We'll explore how sexual reproduction gives organisms a crucial advantage against rapidly evolving parasites, a constant battle where standing still means falling behind. Join us as we uncover why shuffling genes might be nature's best defense in this never-ending race for survival. Chapter 1. The Paradox of Sex. Sex, from an evolutionary perspective, makes very little sense. This might seem like a strange statement, given how ubiquitous sexual reproduction is throughout the natural world. From algae to elephants, from microscopic fungi to towering trees, the vast majority of eukaryotic organisms engage in sexual reproduction. Yet when we examine the process carefully, when we tally up the evolutionary costs and benefits, sex appears to be a remarkably inefficient strategy for passing genes to future generations. This paradox has troubled evolutionary biologists for decades, leading one researcher to call understanding sex the queen of problems in evolutionary biology. To understand why sex presents such a puzzle, we need to consider what it actually costs an organism to reproduce sexually rather than simply making copies of itself. The most dramatic of these costs was identified by evolutionary biologist John Maynard Smith in the early 1970s. He called it the twofold cost of sex, though it's also known as the cost of males. This cost applies specifically when males contribute little or nothing to offspring care beyond their genetic material. Imagine a population of fish living in a lake. These fish reproduce sexually, with roughly equal numbers of males and females. Consider a female who produces exactly 2 offspring during her lifetime. Given equal investment in male and female offspring, we would expect one of these to be male and one to be female. Now let's follow what happens to this female's genetic legacy. Her daughter will also produce 2 offspring during her lifetime. So our original female ends up with 2 grandchildren. This seems reasonable enough. Now imagine that a mutation arises that allows a female to reproduce asexually, producing daughters that are clones of herself. This asexual female also produces 2 offspring during her lifetime. But here's the critical difference. Both of her offspring are female, and both of them can reproduce directly. Each daughter produces 2 more offspring. Our asexual female now has 4 granddaughters compared to the 2 grandchildren of her sexual cousin. An asexual female's lineage doubles in frequency each generation relative to sexuals, a devastating demographic advantage. Within a relatively short time, assuming all else equal, the asexual clone should completely replace the sexual population. The mathematics here are ruthlessly simple. Sexual females invest half of their reproductive resources in producing males. These males, while necessary for reproduction in a sexual population, do not themselves bear offspring. From a strictly demographic perspective, they represent wasted reproductive potential. An asexual female, by contrast, produces only daughters, who all contribute directly to population growth. This gives asexual reproduction a potentially overwhelming advantage. This cost isn't just theoretical. Consider the New Zealand freshwater snail, Potema purgus antipodarum. Populations of this species include both sexual individuals and asexual clones. In the absence of other selective pressures, the asexual clones should rapidly outcompete their sexual counterparts through this simple arithmetic advantage. Yet, sexual reproduction persists in these populations, suggesting that something must be counterbalancing the enormous demographic cost. But the situation becomes even more complicated when we consider organisms where males do contribute substantially to parental care. In such species, there isn't a full twofold cost of males, because males help rear offspring and improve their survival. Yet even these species face another fundamental problem with sexual reproduction, one that affects all sexual organisms regardless of male parental investment. This is what evolutionary biologist George Williams called the cost of meiosis. The cost of meiosis is fundamentally different from the cost of males. While the cost of males is demographic, about population growth rates, the cost of meiosis is genetic. When organisms reproduce asexually, they pass on their complete genome to their offspring. The genetic relationship between parent and offspring is 100%. But in sexual reproduction, meiosis reduces this by half. Each offspring receives only 50% of each parent's genes. From the gene's eye view of evolution, this represents a significant dilution of genetic representation in the next generation. An asexual parent achieves twice the genetic representation in its offspring compared to a sexual parent. This cost applies whether or not males provide parental care. But the costs of sex extend beyond these genetic and demographic considerations. There's also recombination load. Genes don't operate in isolation. They work together in intricate networks, and evolution shapes combinations of genes that function well together. An individual that has survived to reproductive age has demonstrated not just that it possesses good genes, but that these good genes work effectively in combination. The organism represents a tested, successful genetic package. This cost applies whether or not males provide parental care. But the costs of sex extend beyond these genetic and demographic considerations. There's also recombination load. Genes don't operate in isolation. They work together in intricate networks, and evolution shapes combinations of genes that function well together. An individual that has survived to reproductive age has demonstrated not just that it possesses good genes, but that these good genes work effectively in combination. The organism represents a tested, successful genetic package. Sexual reproduction, through the process of recombination, potentially breaks apart these successful gene combinations. When chromosomes exchange segments during meiosis, alleles that worked well together may be separated. New combinations are created, but there's no guarantee these will work as effectively as the parental combinations. For organisms finely tuned to their environment, sexual reproduction introduces the risk of disrupting advantageous genetic arrangements. An asexual parent, by contrast, passes on its successful genotype intact to all offspring. The costs don't end there. Sex requires finding a mate, and this process itself consumes considerable time and energy that could otherwise be devoted to reproduction or survival. Consider the elaborate courtship displays observed throughout the animal kingdom. Male birds of paradise perform intricate dances. Male frogs call through the night. Male deer engage in spectacular battles with their antlers. These activities require enormous energy expenditure. They also expose organisms to predators. A singing frog announces its location not just to potential mates, but to any predator within earshot. The peacock's tail provides perhaps the most famous example of the costs associated with sexual reproduction. This enormous ornament is a serious survival handicap. It requires substantial energy to grow and maintain. It makes the male more visible to predators and more difficult to maneuver when escaping danger. If male peacocks could reproduce asexually, they would have no need for such elaborate displays. All that energy could be directed toward producing more offspring. Yet not only does the tail exist, it continues to evolve toward ever greater elaboration through female choice. Sexual reproduction also brings risks that asexual reproduction avoids entirely. Mating brings organisms into close physical contact, creating opportunities for disease transmission. Sexually transmitted infections represent a class of parasites that exploit the very act of reproduction. An asexual organism faces no such risks. It need never come into contact with another member of its species. Even at the cellular level, sex is more expensive. Meiosis, the specialized cell division required for sexual reproduction, takes considerably longer than mitosis, the simple cell division used in asexual reproduction. This additional time represents resources that could be devoted to other fitness-enhancing activities. Interestingly, some organisms have evolved an intermediate strategy that partially mitigates these costs while retaining some benefits. Self-fertilization, or selfing, allows organisms to reproduce sexually without requiring a mate. This reduces the cost of sex by eliminating mate-finding expenses and allowing organisms to produce only offspring-bearing individuals. However, selfing also reduces the genetic diversity benefits of sexual reproduction by creating extensive homozygosity. A selfing individual essentially mates with itself, producing offspring that are much more genetically similar to the parent than outcrossed offspring would be. Selfing represents a compromise between full sexual reproduction and asexuality, reducing both the costs and the benefits. Given this daunting list of costs, the demographic expense of producing males, the genetic cost of reduced relatedness to offspring, the breakup of successful gene combinations, the energy spent finding and competing for mates, the risk of disease transmission, and the time required for the sexual cell cycle, we arrive at a profound puzzle. Why does sex dominate among eukaryotes? What benefits could possibly outweigh such substantial costs? The answer, supported by decades of research, appears to lie in the relentless pressure of parasites and pathogens. The benefit that counterbalances all these costs is the genetic diversity that sexual reproduction creates among offspring. This diversity becomes crucial when organisms face rapidly evolving enemies. Parasites pose a unique evolutionary challenge. They typically have much shorter generation times than their hosts. A bacterium can complete its life cycle in hours, producing hundreds of offspring. A virus can replicate thousands of times within a single host infection. These microscopic enemies evolve far more rapidly than their hosts, potentially adapting to common host genotypes within relatively few host generations. In an asexual population, all individuals share the same genotype. Once a parasite evolves the ability to infect one individual, it can potentially infect the entire population with equal effectiveness. The population presents a uniform target. As the most successful asexual genotype increases in frequency, parasites adapted to that genotype also increase, eventually driving the common clone to low frequency or extinction. Sexual reproduction offers an elegant solution to this problem. By recombining genes each generation, sexual organisms produce offspring that differ genetically from their parents and from each other. Parasites that have adapted to infect the parental generation may find themselves poorly equipped to infect the diverse array of offspring genotypes. Sexual reproduction essentially presents parasites with a moving target, a constantly shifting landscape of host genetics. Evidence for this comes from elegant experiments with the crustacean Daphnia magna and its bacterial parasite Pasturia ramosa, detailed in Chapter 3, which showed that sexually produced offspring were twice as resistant to infection as their asexual siblings when facing co-evolved parasites. Even more dramatic evidence comes from experiments with the microscopic roundworm Caenorhabditis elegans. These worms can reproduce either sexually or by self-fertilization. Researchers genetically engineered populations that could only reproduce asexually and others that could only reproduce sexually. They then exposed these populations to Serratia marcescens, a bacterial pathogen that invades the worm's body and can kill it in under 24 hours. The asexual populations that faced co-evolving bacteria went extinct within a mere 20 generations, a remarkably short time representing just weeks in the laboratory. The bacteria became more and more effective at killing these genetically uniform hosts, and the hosts had no way to evolve new defensive strategies quickly enough. In contrast, sexual populations persisted throughout the experiment. They maintained resistance despite facing the same evolving pathogen. The key difference was the constant generation of new genetic combinations through sexual reproduction. Critically, when researchers exposed worms to frozen bacteria that couldn't evolve, even asexual populations survived. This proved that the crucial factor wasn't simply the presence of parasites, but the presence of parasites that were themselves evolving. Only when both host and parasite were locked in a coevolutionary struggle did sexual reproduction provide a decisive advantage. Studies of wild populations reinforce these laboratory findings. In New Zealand, mud snails living in shallow lake waters where parasitic trematode worms are common, sexual reproduction predominates. In deeper waters of the same lakes, where ducks don't forage and therefore can't transmit the parasite, asexual clones dominate. The spatial distribution of sexual versus asexual reproduction tracks precisely with parasite presence. The magnitude of the benefit that sex provides becomes clear when we consider what's required to overcome the twofold cost. For sexual reproduction to be evolutionarily stable, it must provide at least a twofold fitness advantage. The experimental evidence showing that sexual offspring can have twice the resistance of asexual offspring to co-evolved parasites demonstrates exactly this magnitude of benefit. This balance between costs and benefits reveals when each reproductive strategy succeeds. In environments with low parasite pressure, stable conditions, or slow-evolving parasites, asexuality can prevail. The demographic advantage overwhelms any modest benefits from genetic diversity. We see this in some Arctic and Antarctic environments, in isolated habitats with depopulate parasite communities, and in recently colonized areas where parasite populations haven't yet built up. Conversely, in environments with high parasite diversity, rapidly evolving parasites, or strong parasite-mediated selection, sexual reproduction dominates despite its costs. The genetic diversity it generates provides immediate fitness benefits large enough to justify the twofold demographic expense. Most natural environments fall into this category, which explains why sex is so widespread. The tipping point between these strategies depends on the strength of parasite-mediated selection. When parasites track common host genotypes rapidly and effectively, as demonstrated in the C. elegans experiments, sex becomes essential. When parasite pressure relaxes or stabilizes, asexuality gains ground. This creates a dynamic tension that has shaped the evolution of reproductive strategies across the tree of life. Understanding this trade-off provides the foundation for exploring the deeper mechanisms of host-parasite coevolution. Every sexually reproducing organism represents a lineage that has, generation after generation, found the benefits of genetic diversity to outweigh the enormous costs of sexual reproduction. The question is not why sex exists, but rather, what exactly are parasites doing to make genetic diversity so valuable? The following chapters explore the coevolutionary dynamics that answer this question. Chapter 2. Running to Stand Still. In Lewis Carroll's Through the Looking Glass, Alice finds herself in a peculiar land where she must run as fast as she can just to stay in the same place. The Red Queen explains to a bewildered Alice, "Now here, you see, it takes all the running you can do to keep in the same place." This seemingly absurd scenario, published in 1871, would eventually give its name to one of the most important concepts in evolutionary biology. A century later, evolutionary biologist Lee Van Valen recognized that this paradoxical state of constant motion without progress perfectly describes the relationship between hosts and their parasites. Van Valen's insight emerged from studying patterns in the fossil record. Examining survivorship curves, graphs showing the probability of extinction over time across diverse taxonomic groups, including bivalves, gastropods, and mammalian families, he noticed something remarkable. The probability of extinction appeared independent of how long a species had existed. Whether a species was young or ancient made no difference to its risk of going extinct. A species that had persisted for 10 million years was just as likely to vanish as one that had appeared only 1,000,000 years ago. This pattern, which Van Valen called the law of constant extinction, demanded explanation. If species were simply adapting to their physical environment, we would expect older species to be better adapted and therefore more resistant to extinction. Yet this wasn't what the fossil record showed. Van Valen proposed a radical solution. The effective environment of any organism, he argued, deteriorates at a constant rate, not because of changes in climate or geology, but because of evolution in competing and parasitic species. Each improvement in one species represents a deterioration in the environment experienced by the species it interacts with. A prey species that evolves to run faster makes the environment worse for its predator. A parasite that evolves improved infectivity makes the environment worse for its host. These changes don't accumulate towards some stable optimum. Instead, they create a perpetual treadmill of evolutionary change where no species gains a lasting advantage. The idea was so controversial that Van Valen's original 1973 paper was rejected by traditional journals. Undeterred, he founded his own journal, Evolutionary Theory, specifically to publish this work. The resistance he encountered reflected how revolutionary his thinking was. Biologists had grown accustomed to viewing evolution primarily as adaptation to physical environments, not as an endless race between species. This is the essence of the Red Queen hypothesis. In biotic interactions, evolution is fundamentally a zero-sum game. The fitness gained by one party equals the fitness lost by the other. When a host population evolves increased resistance to a parasite, the parasite's average fitness necessarily declines. When the parasite evolves to circumvent that resistance, the host's average fitness drops back down. Both species evolve continuously, yet neither gains lasting advantage over the other. They run faster and faster, yet remain locked in the same relative positions. Stop evolving, even for a moment, and you fall behind. This stands in stark contrast to evolution in response to the abiotic environment. When organisms adapt to temperature, rainfall, or soil chemistry, they can reach an optimal state and remain there as long as physical conditions stay constant. A cactus adapted to desert conditions doesn't need to continue evolving its water storage capacity once it has achieved an efficient design. The desert doesn't evolve back. But parasites do evolve, and this fundamental difference creates entirely different evolutionary dynamics. The mechanism driving Red Queen dynamics operates through negative frequency-dependent selection. The most common host genotype suffers the greatest parasite burden simply by virtue of being common. Parasites in the population have been selected to overcome the defenses of common hosts because that's where the most opportunities for infection exist. But this very success changes the selective landscape. As the common host genotype declines in frequency due to heavy parasite burden, previously rare host genotypes increase. These rare genotypes initially suffer less parasitism because parasites haven't adapted to them. But as they become common, parasites evolve to exploit them, and the cycle begins again. This creates oscillating dynamics in host and parasite populations. Genotype frequencies rise and fall not in response to changes in the physical environment, but in response to each other. The parasite population always lags slightly behind the host population, adapting to yesterday's common host genotype, while today's hosts have already begun shifting toward different genetic variants. Likewise, hosts respond to yesterday's common parasite genotype. This perpetual lag, this constant chase where neither party can quite catch the other, drives continuous evolutionary change in both species. Evidence for these cyclical dynamics comes from careful long-term monitoring of natural populations. In a large population of Daphnia magna living in Lake Eigelsee in Switzerland, researchers Jokela Jokela, Mark Dibdahl, and Curtis Lively tracked the genetic composition of both host and parasite from October 2010 through September 2018. Every spring, Daphnia emerge from sexually produced resting eggs that have overwintered in the lake sediment. Throughout the summer, these water fleas reproduce asexually, creating clonal lineages that can be tracked individually. As summer progresses, epidemics of the bacterial parasite Pasteurella ramosa sweep through the population, with infection rates reaching nearly 100%. The researchers collected samples every few weeks throughout each growing season, cloning individual Daphnia and testing their resistance to parasites. They discovered a remarkable pattern. Early in the season, when Daphnia first emerged from their resting eggs, the population showed high genetic diversity and resistance. Many different resistance genotypes coexisted. As the summer epidemic progressed, this diversity collapsed. The most resistant genotypes increased in frequency, while susceptible genotypes declined. Picture the surface of the lake over the course of a summer. In May, a diverse mixture of clonal lineages multiplying in the warming water. By July, the population shifting visibly as certain clones dominate while others fade. By September, the lake populated primarily by descendants of the few lineages that could withstand that year's parasite onslaught. By late summer, the population had become dominated by the resistant genotypes that could best withstand that year's parasite strain. But here's the crucial observation. When the population emerged again the following spring from sexually produced resting eggs, genetic diversity for resistance was restored. The highly resistant population from the previous fall produced offspring whose resistance varied widely, including many susceptible individuals. Sexual reproduction had shuffled the genetic deck, recreating the variation that selection during the epidemic had eliminated. And the cycle began anew. Each year, selection during the epidemic favored resistance. Each spring, sex recreated susceptibility. This isn't simply random variation being regenerated. The pattern reveals something deeper about the genetic architecture of resistance. When highly resistant parents reproduce sexually, their offspring include many susceptible individuals. This can only happen if resistance involves complex genetic interactions where specific combinations of alleles are required for the resistance phenotype. Sexual reproduction breaks apart these successful combinations, producing offspring with new arrangements of resistance alleles, some of which confer much less protection than the parental combinations. From the parasite's perspective, the dynamics are equally dramatic. Researchers examining the same Daphnia population over multiple years found that parasites collected early in the epidemic were less infectious than parasites collected later. Within a single season, the parasite population evolved increased infectivity. But this evolution was specific. Parasites became better at infecting the host genotypes present that year. When researchers tested parasites against host genotypes from previous years, the parasites were often less effective, demonstrating that parasite evolution tracked changes in host genetics rather than simply becoming universally more virulent. The temporal dynamics can be extended even further backward using a remarkable property of some aquatic systems. In stratified lakes, sediments accumulate in distinct annual layers. The resting eggs of Daphnia, along with the resting spores of their parasites, become entombed in these layers, creating a biological archive. By carefully extracting sediment cores and reviving these resting stages, researchers can resurrect hosts and parasites from decades past. Studies using this resurrection approach have demonstrated coevolution over timescales of decades. Parasites from a particular sediment layer were most infectious against hosts from that same layer. They were less effective against hosts from earlier layers and from later layers. This pattern indicates ongoing coevolutionary dynamics. Parasites continuously adapt to contemporary hosts, becoming specifically tuned to the genotypes present at that moment in time. When host populations shift genetically through either selection or sexual reproduction, parasites must evolve anew to maintain their infectivity. Direct evidence for this constant evolutionary motion comes from experiments that frozen host and parasite populations at different time points and then tested all possible combinations. A parasite from year 1 tested against hosts from year 1 shows moderate infectivity. The same parasite tested against hosts from year 3 often shows lower infectivity because the hosts have evolved. But here's the remarkable part. Hosts from year 3 tested against parasites from year 3 show similar levels of infection as the year 1 combination. Both parties have evolved, but neither has gained lasting ground. They've both been running, and both remain in essentially the same place relative to each other. Even more dramatic evidence comes from experiments with the tiny nematode Canorhabditis elegans and its bacterial parasite Serratia marcescens. Researchers maintained populations of sexual and asexual worms under identical conditions except for their mode of reproduction, then exposed both to the evolving parasite. The asexual worm populations went extinct within 20 generations, unable to generate sufficient variation to keep pace with the rapidly adapting bacteria. The sexual populations persisted indefinitely. The Red Queen's race proved fatal for those who couldn't shuffle their genetic deck. The dynamics of Red Queen coevolution don't proceed uniformly across all environments. Instead, different populations of the same host and parasite species may undergo different coevolutionary trajectories depending on local conditions. This geographic mosaic of coevolution creates spatial variation in the outcome of host-parasite interactions. Consider the powdery mildew Potosphaera plantagenus and its plant host Plantago lanceolata, studied across thousands of populations in the Åland Islands of southwestern Finland. The mildew obtains nutrients by sending feeding roots into the plant tissue. Both host and parasite can reproduce sexually and asexually, and the intensity of their interaction varies dramatically across the landscape. In some locations, the mildew regularly overwinters and causes intense summer epidemics. In other populations, the parasite frequently goes extinct locally during winter, creating areas where hosts experience little parasitism. These differences in parasite pressure create different selective environments. In populations where parasites are consistently present and causing epidemics, strong reciprocal selection drives rapid coevolution. Resistant plant genotypes increase in frequency. Parasites evolve to overcome this resistance, and the cycle continues. But in populations where parasites are frequently absent, there's little selection for resistance, and the host population can drift genetically or be shaped primarily by other selective forces. When parasites eventually arrive in such populations, they may encounter hosts that are highly susceptible because selection for resistance has been relaxed. This geographic variation has profound consequences. Different populations become adapted to their local parasites and hosts. A parasite that is highly infectious in one location may be completely ineffective in another, not because the hosts are different species, but because they've taken different coevolutionary paths. The populations form a spatial mosaic where coevolution proceeds at different rates and in different directions, depending on local ecological conditions. The Red Queen framework also helps explain why neither hosts nor parasites evolve to a final, stable state. We might imagine that eventually hosts would evolve perfect resistance, or parasites would evolve perfect infectivity. But this doesn't happen. Perfect resistance would require hosts to defend against all possible parasite strategies simultaneously, which is likely impossible and certainly expensive. Instead, hosts evolve resistance to the specific parasite genotypes they currently encounter, leaving them vulnerable to other parasite strategies. When parasites evolve to exploit these vulnerabilities, hosts must shift their resistance profile, abandoning defenses against yesterday's parasites to combat today's threats. From the parasite's perspective, perfect infectivity is similarly elusive. The parasite faces a constantly changing target as host populations shift genetically. Moreover, evolution of increased infectivity often comes with costs. A parasite that replicates extremely rapidly might kill its host before transmission can occur. A parasite that triggers strong immune responses might be efficiently cleared. The optimal parasite strategy depends on the current state of the host population, and because that state is always changing, the optimal parasite strategy is always changing too. This perpetual motion has important consequences for the maintenance of sexual reproduction. In a stable environment where hosts could evolve to a fixed optimum and remain there, sexual reproduction's shuffling of genes would be disadvantageous. Once a successful gene combination arose, it would be better to preserve it through asexual reproduction. But in the constantly changing selective landscape created by co-evolving parasites, no gene combination remains optimal for long. Today's successful combination becomes tomorrow's target for parasite adaptation. By continuously creating new genetic combinations, sexual reproduction prevents any single host genotype from becoming common enough to be efficiently tracked by parasites. The strength of selection required to maintain sex can be remarkably intense. In the Daphnia populations experiencing annual epidemics, resistant genotypes can increase from low frequency to dominance within a single season. This represents selection coefficients far stronger than those typically observed in most natural populations. Yet even this strong selection during the asexual phase of reproduction is counterbalanced by the restoration of variation through sex. The system remains in constant flux, with neither extreme resistance nor extreme susceptibility dominating for long. This constant evolutionary motion distinguishes red queen dynamics from other forms of frequency-dependent selection. In some ecological interactions, frequency dependence can lead to stable equilibria where multiple genotypes coexist indefinitely without ongoing change in their frequencies. But in host-parasite interactions with genetic specificity, the system rarely, if ever, settles into stasis. The lag between parasite adaptation and host evolution, combined with the continuous generation of new variation through mutation and recombination, keeps the system in perpetual motion. The red queen hypothesis thus provides a mechanism for continuous, rapid evolution in the face of apparently unchanging environmental conditions. A lake might show little variation in temperature, chemistry, or physical structure over decades, yet the species within it continue evolving at a rapid pace driven by their interactions with each other. This ongoing evolution, invisible in the traditional fossil record that focuses on morphology, represents the majority of evolutionary change occurring in natural populations. Understanding these red queen dynamics illuminates why sex persists despite its costs. Sexual reproduction is not simply a mechanism for occasional adaptation when environments change. Rather, it is the essential tool for keeping pace in a race that never ends. The parasite will evolve. The host must therefore evolve. And the only way to evolve quickly enough in a constantly changing biotic landscape is to continually generate new genetic combinations through sex. The Red Queen's realm is one of constant motion, where survival requires running faster and faster just to stay in the same place. Chapter 3. Racing Against Evolution. Testing the Red Queen hypothesis requires more than observing patterns in nature. To truly understand whether parasites drive the evolution and maintenance of sexual reproduction, scientists needed experiments that could directly manipulate host reproductive strategies while tracking parasite evolution in real time. The freshwater crustacean Daphnia magna and its devastating bacterial parasite Pasteurella ramosa provided the perfect system for such experiments. The unique biology of this host-parasite pair allowed researchers to conduct tests that would be impossible in most other organisms. Daphnia are tiny crustaceans, barely visible to the naked eye, that inhabit ponds and lakes throughout the northern hemisphere. Their reproductive biology makes them extraordinarily useful for studying the evolution of sex. During favorable conditions in spring and summer, Daphnia females reproduce asexually, releasing live-born daughters that are genetic clones of their mother. These clonal lineages can be maintained indefinitely in the laboratory, creating large populations of genetically identical individuals. But when conditions deteriorate, or when population density rises, Daphnia switch to sexual reproduction, producing males and special resting eggs called ephippia. These ephippia are encased in a protective saddle-shaped structure and can survive freezing, desiccation, and passage through the digestive systems of waterfowl. In temperate regions, ephippia allow Daphnia to survive winter, hatching when conditions improve in spring. These resting eggs have made Daphnia central to a field called resurrection ecology. Ephippia accumulate in pond sediments year after year, creating natural archives of genetic diversity. By extracting sediment cores and hatching ephippia from different layers, researchers can literally resurrect populations from decades or even centuries past. This has allowed scientists to study evolution in real time by comparing historical populations to their modern descendants. The time-shift experiment that Stuart Old, Shona Tinkler, and Matthew Tinsley designed built on this tradition but compressed the timescale dramatically. Rather than comparing populations separated by decades, they would compare parasites separated by just 1 year of evolution. Pasteurella ramosa is equally remarkable, though far more sinister from the Daphnia's perspective. This bacterium infects Daphnia when the tiny crustaceans filter spores from the water during feeding. Once inside the host's gut, spores germinate and the bacteria penetrate the gut wall, entering the body cavity. From there, Pasteurella spreads throughout the host's tissues, eventually filling the animal with millions of transmission spores. The infection is absolutely sterilizing. An infected female produces no offspring at all. She becomes a walking spore factory, her reproductive system completely shut down by the parasite. When she dies, her body ruptures, releasing millions of spores into the water to infect the next generation of hosts. The bacteria growing inside the host's body cavity give infected individuals a distinctive white or cloudy appearance, easily distinguished from the transparent bodies of uninfected animals. The researchers traveled to a natural pond at Keims Farm in the Scottish borders in June of 2013. The timing was crucial. In June, Daphnia populations in this region are typically in the midst of their transition to sexual reproduction. The researchers collected 52 healthy female Daphnia that were carrying Ephipia. These Ephipia contained sexually produced eggs that had already been fertilized. The researchers also collected 100 infected Daphnia from the same pond. These infected individuals were literally bursting with Pasteurella spores, each containing anywhere from 400,000 to over 6 million transmission spores. This represented the contemporary parasite population evolved to infect the Daphnia genotypes present in the pond that year. Back in the laboratory, the infected Daphnia were homogenized to release their spores. The researchers pooled these spores into a single suspension representing the year 1 parasite population and stored them at -20 degrees Celsius. The healthy females carrying Ephipia were isolated individually and watched carefully. After these females released their Ephipia into the water, something remarkable happened. The same females that had just produced sexual eggs reverted to asexual reproduction. Within days, they began producing clonal daughters. These asexual offspring were collected and used to establish clonal lines representing the maternal genotype of each family. Meanwhile, the Ephipia were treated with a dilute bleach solution to remove any external contamination, then placed individually in small volumes of artificial culture medium positioned on a windowsill to receive natural sunlight, which helps trigger hatching. Out of the 54 collected, 21 eventually hatched. Some Ephipia contained only one viable egg, while others contained 2. In total, the researchers established 21 Daphnia families. Each family consisted of one asexual genotype derived from the mother and either 1 or 2 sexual genotypes derived from the offspring that hatched from that Ephipium. This experimental design was elegant in its simplicity. Within each family, the asexual genotype represented what would happen if the mother simply cloned herself. The sexual genotypes represented what would happen if she reproduced sexually instead. Any differences in parasite resistance between sexual and asexual offspring from the same family had to result from the shuffling of genes during sexual reproduction, not from differences in which genes were available. But creating these families was only the first step. The researchers needed to ensure that all the Daphnia were in comparable physiological condition before testing their parasite resistance. To control for potential environmental and maternal effects, the researchers maintained 8 independent replicate cultures of each Daphnia genotype for 3 complete generations under standardized conditions. After 3 generations, the researchers collected second clutch offspring from the 3rd generation to use in the actual experiment. This choice mattered because Daphnia often showed differences in offspring quality between their first and subsequent clutches, with later clutches typically being more vigorous. By using second clutch offspring from the 3rd generation, the researchers ensured that any lingering maternal effects from the original wild-caught females had been eliminated and that all individuals were in peak condition. 1 year after the initial collection, in June of 2014, the researchers returned to the same pond at Keims Farm. They collected another 100 infected Daphnia. These were homogenized just like the previous year's collection, and the spores were pooled to create the year 2 parasite population. This collection represented parasites that had evolved in the natural pond for 1 additional year, potentially adapting to host genotypes that had become common during that time. This was the critical feature of the experiment. If parasites adapt to common host genotypes, then year 2 parasites should be better at infecting asexual clones, which would have remained genetically identical to their mothers. But sexual offspring, with their shuffled genetic combinations, might present novel targets that the evolved parasites hadn't encountered. Now came the critical experiment. The researchers divided the offspring from each family into 2 groups using a split clutch design. One group would be exposed to year 1 parasites, collected at the same time as their mothers. The other group would be exposed to year 2 parasites, representing parasites that had evolved for an additional year in the wild. For each combination of host genotype and parasite year, the researchers set up 8 replicate cultures with 8 individual Daphnia each. The parasite exposure protocol was precisely controlled. Each replicate received exactly 100,000 Pastorius spores, either from the year 1 or year 2 collection. During the 5-day exposure period, the cultures were stirred daily to keep spores suspended, and the Daphnia were fed reduced rations to ensure they would filter more actively. After 5 days of exposure, the Daphnia were transferred to fresh medium free of spores and returned to normal feeding. The experiment continued for 30 days total. 20 days after the initial exposure, the researchers examined each culture and visually scored which individuals had become infected based on their distinctive white, cloudy appearance. At the end of the 30-day experiment, the researchers pooled all the Daphnia from each replicate culture and counted the number of Pastorius spores they contained, providing a measure not just of infection rate, but of infection intensity. The results were striking. When exposed to year 1 parasites, both sexual and asexual offspring showed similar infection rates, averaging around 40% across families. This made sense. The year 1 parasites had been collected at the same time as the mothers of these experimental Daphnia. They represented the contemporary parasite population against which natural selection had been acting. Neither sexual nor asexual offspring had any special advantage against these parasites. But when the researchers exposed the same host genotypes to year 2 parasites, a dramatic difference emerged. Asexual offspring showed infection rates averaging around 55% when exposed to the evolved parasites, a significant increase compared to year 1 parasites. The parasites had adapted during their additional year in the wild, and this adaptation had made them more effective at infecting these asexual genotypes. The asexual hosts, being genetic clones of their mothers, presented the same target to parasites as the previous generation had. If parasites in the wild had adapted to infect that target, they would be more successful. Sexual offspring showed a completely different pattern. They maintained infection rates around 30% when exposed to year 2 parasites, showing no increase in susceptibility to the evolved parasites. When directly compared to their asexual siblings exposed to the same year 2 parasites, sexual offspring were approximately twice as resistant. This wasn't a subtle difference. It was a dramatic protection that matched almost perfectly the twofold demographic cost that sexual reproduction imposes. Sexual reproduction didn't provide a slight advantage. It provided an advantage large enough to fully compensate for its enormous cost. The spore density data told an equally compelling story. When asexual hosts did become infected by year 2 parasites, they contained significantly higher spore densities, often exceeding 2 million spores per individual, compared to asexual hosts infected by year 1 parasites, which typically contained around 1 million spores. The parasites hadn't just become better at establishing infections, they'd become better at replicating within asexual hosts, producing more transmission spores per infected individual. Sexual hosts, by contrast, showed no such increase in parasite replication with the evolved parasites. When sexual offspring did become infected, they contained similar spore densities regardless of whether parasites came from year 1 or year 2. The genetic shuffling that occurred during sexual reproduction had apparently created combinations of resistance genes that the parasites hadn't encountered before. These results demonstrated several critical points about how the red queen dynamic actually operates in nature. First, parasites can evolve rapidly, adapting to common host genotypes within a single year. This is far faster than many had suspected. Previous resurrection ecology studies had shown evolution over decades, but this experiment revealed that the arms race could proceed at breakneck speed when parasite generations are short and selection is intense. Second, this evolution is specifically directed at the genotypes actually present in the host population. The year 2 parasites weren't just generally more virulent, they were specifically better at infecting the asexual genotypes that would have been present in the natural population, descendants of the same mothers that had produced the Ephippia. Third, sexual reproduction provides immediate and substantial protection against these adapted parasites by creating novel genetic combinations. The experimental design eliminated many alternative explanations. Because sexual and asexual offspring came from the same mothers, differences in resistance couldn't be attributed to which genes were present. The genes were the same. What differed was how they were combined. Because all Daphnia were raised for 3 generations under identical laboratory conditions, differences couldn't be attributed to environmental effects or maternal condition. Because the exposure protocols were rigorously standardized, differences couldn't be attributed to variation in parasite dose or environmental conditions during infection. What makes these results particularly significant is that they demonstrate the Red Queen hypothesis operating at a timescale relevant to actual population dynamics. A 1-year advantage is not some distant evolutionary benefit that accrues over geological time. It's an immediate, generation-by-generation advantage that can maintain sexual reproduction in populations where asexual mutants constantly arise. When an asexual clone appears in a sexual population, it should initially thrive, producing twice as many offspring as its sexual competitors. But within a single year, parasites adapt to that common clone, reducing its fitness. Meanwhile, sexual individuals continue producing diverse offspring, staying one step ahead in the evolutionary race. The magnitude of the benefit is equally important. Theoretical models had long predicted that parasites would need to impose very strong selection to overcome the twofold cost of sex. The Daphnia pastoria system demonstrated exactly this kind of strong selection in action. A twofold difference in infection rate, combined with the complete sterilization imposed by Pastoria, meant that asexual clones could go from having a twofold reproductive advantage to having near zero fitness within a single year if parasites were abundant. This creates precisely the kind of fluctuating selection that Red Queen models require. The Red Queen hypothesis, long supported by theoretical models and correlational evidence, now had direct experimental confirmation. Parasites adapt rapidly to common host genotypes. Sexual reproduction protects against this adaptation by creating rare genetic combinations. The benefit is both large enough and immediate enough to overcome the substantial costs of sexual reproduction. Sex persists not despite its costs, but because parasites make those costs worthwhile. In the evolutionary race between hosts and parasites, standing still means falling behind, and the only way to keep running is to shuffle the genetic deck with every generation. Chapter 4. The Architecture of Coevolution. The previous chapters have established that parasites can favor sexual reproduction and that hosts and parasites engage in continuous coevolution. But these broad patterns obscure a more intricate reality. Host-parasite coevolution doesn't proceed uniformly or predictably. Instead, it unfolds through a complex interplay of rapid evolutionary responses, shifting genetic architectures, and fluctuating selection pressures that vary across time and space. To understand how parasites truly shape host evolution, we must examine the detailed mechanisms of these reciprocal adaptations. The speed at which hosts and parasites can evolve in response to each other often surprises researchers who are accustomed to thinking of evolution as a slow process requiring millions of years. In many host-parasite systems, meaningful evolutionary change occurs within tens of generations or even fewer. This rapidity stems from the intense selection that parasites impose on hosts and that hosts impose on parasites in return. When a bacterial parasite can reduce host fitness to 0 through complete sterilization, any genetic variant that provides even partial resistance experiences enormously strong selection. Similarly, when a host population evolves resistance that blocks 99% of infections, parasites carrying mutations that restore infectivity enjoy dramatic fitness advantages. Consider the dynamics observed in populations of the bacterium Escherichia coli and the viruses that infect them, known as bacteriophages. In laboratory evolution experiments, researchers can watch these interactions unfold in real time. A phage population begins with the ability to infect bacterial cells by binding to a specific receptor on the cell surface. This receptor normally serves important metabolic functions for the bacterium, but it creates a vulnerability that phages exploit. Within days, bacteria carrying mutations that alter or eliminate this receptor appear in the population and rapidly increase in frequency. These mutants are resistant to the original phage strain. The resistance comes at a cost. Mutations that alter the receptor to prevent phage binding often reduce the receptor's normal metabolic function. Resistant bacteria may grow more slowly than their susceptible ancestors when phages are absent, creating a trade-off between resistance and baseline fitness. This illustrates a fundamental constraint on the evolution of defense. Protective mutations often impose pleiotropy costs, affecting other traits beyond their defensive function. But the phages don't simply go extinct. Within a similar time frame, phage mutants arise that can bind to the altered receptor or to a different receptor entirely. These counter-adapted phages can again infect the resistant bacteria, and they spread through the phage population. The cycle continues with bacteria evolving new resistance mechanisms and phages evolving new ways to circumvent them. Across just a few weeks of laboratory culture, researchers can observe multiple rounds of adaptation and counter-adaptation, each driven by the previous evolutionary move of the antagonist. The genetic basis of infection and resistance profoundly influences the patterns of coevolution that emerge. At one extreme lies what researchers call gene-for-gene dynamics. This system, first discovered in agricultural plants and their fungal pathogens, involves matching between specific host resistance genes and specific parasite infectivity genes. A host carries resistance gene R1, which provides protection against parasites carrying infectivity gene I1. But this resistance offers no protection against parasites carrying a different infectivity gene, I2. Similarly, a parasite carrying I2 can infect hosts with R1, but not hosts carrying resistance gene R2. This matching creates a peculiar form of selection. Common host genotypes become targets for parasite adaptation. Parasites carrying the infectivity genes that can overcome the common resistance genes spread through the parasite population. But as these parasites increase in frequency, they shift selection on the host population, favoring rare resistance genes that the common parasites cannot overcome. The formerly common host genotypes decline, rare genotypes increase, and the identity of what is common and rare flips. This drives oscillations in genotype frequencies in both populations. The Daphnia magna pastoria system from Chapter 2 revealed a crucial genetic mechanism underlying these cycles. The genetic architecture of resistance involves interactions among multiple genes. Specific combinations of alleles at different loci are required to produce the resistant phenotype. This genetic complexity has crucial consequences. When two resistant parents reproduce sexually, the genetic shuffling that occurs during meiosis breaks apart the specific combination of alleles that made the parents resistant. Many offspring receive incomplete sets of the necessary alleles and are therefore susceptible despite having resistant parents. This phenomenon, called genetic slippage, explains why sexual reproduction can restore susceptibility to populations that had become highly resistant through selection. It isn't that new mutations are creating susceptible genotypes. Rather, sexual recombination is breaking apart the multi-locus genotypes required for resistance, creating offspring with new allelic combinations that don't confer the same level of protection. The population slips backward genetically, not to some primordial state, but to a state where resistance varies widely because the genetic basis of resistance has been reshuffled. The underlying mechanism involves epistasis, where the effect of an allele at one locus depends on which alleles are present at other loci. Imagine that full resistance requires specific alleles at 3 different genes. An individual carrying the resistant allele at only 1 or 2 of these genes might be just as susceptible as an individual carrying none of them. Only the particular combination of all 3 resistance alleles produces the resistant phenotype. When resistant carents reproduce sexually, their offspring receive random combinations of alleles from each parent. Some get all 3 resistance alleles, others get 2, or 1, or none. The population average resistance therefore declines, even though no individual alleles have changed frequency. This genetic architecture creates a persistent tension between selection during the asexual phase of reproduction and recombination during the sexual phase. Selection during clonal reproduction drives specific multi-locus genotypes to high frequency. Sexual reproduction breaks these genotypes apart, recreating variation that selection then acts upon again. Neither process wins permanently. The cycle repeats indefinitely. Not all coevolutionary dynamics follow this pattern. Researchers have identified 3 distinct modes that host parasite systems can exhibit, each characterized by different underlying genetics and selection regimes. The fluctuating red queen mode, which includes the matching dynamics described above, occurs when resistance and infectivity depend on gene for gene or multi-locus matching. Genotype frequencies oscillate over time as selection favors rare variants, maintaining high genetic diversity through negative frequency-dependent selection. The fluctuating red queen mode, which includes the matching dynamics described above, occurs when resistance and infectivity depend on gene for gene or multi-locus matching. Genotype frequencies oscillate over time as selection favors rare variants, maintaining high genetic diversity through negative frequency-dependent selection. The escalatory red queen mode represents a different dynamic where both parties evolve along quantitative axes of escalation. Here, resistance and infectivity are not about specific matching, but about threshold levels. A host with higher resistance value can resist parasites below a certain infectivity threshold. A parasite with higher infectivity can overcome hosts below a certain resistance threshold. Evolution proceeds directionally. Hosts evolve ever higher resistance, parasites evolve ever higher infectivity. Both parties escalate their defenses and offenses in an ongoing competition. This mode tends to erode genetic diversity as directional selection favors extreme values. The chase red queen mode occurs in systems with high mutation rates where new beneficial mutations arise faster than selection can fix them. Rather than oscillating genotype frequencies or directional escalation, populations are constantly tracking shifting fitness peaks. This mode is common in RNA viruses, where extremely high mutation rates generate continuous streams of new variants. Within a single infected individual, the virus population diversifies rapidly, and the immune system chases these variants, selecting for escape mutants that avoid recognition. The distinction between these modes has important consequences for how diversity is maintained and how predictable coevolutionary trajectories are. The fluctuating mode actively maintains standing variation. The escalatory mode consumes variation but may be constrained by trade-offs. The chase mode generates variation constantly through mutation rather than preserving it through balancing selection. Trade-offs between different components of parasite fitness can constrain escalatory dynamics. In the flower beetle Tribolium and its microsporidian parasite Nosema, high transmission rates come at the cost of reduced within-host replication, while high virulence that maximizes within-host growth reduces transmission efficiency. Parasites cannot simultaneously maximize all fitness components, creating a constraint on how far escalation can proceed. Such trade-offs can stabilize coevolutionary dynamics by preventing either party from achieving complete dominance. Real host parasite systems often show elements of multiple modes. The interaction between snails and schistosome parasites illustrates this complexity. Schistosomes are flatworms that cause serious disease in humans but require freshwater snails as intermediate hosts. Different schistosome strains show varying degrees of host specificity. Some can infect only particular snail species or populations, suggesting gene-for-gene type matching. But within compatible combinations, there's also variation in how successfully the parasite replicates, suggesting quantitative variation in resistance and infectivity that could fuel escalatory dynamics. The temporal pattern of selection also varies across systems. In some host-parasite interactions, selection intensities fluctuate wildly over time. During epidemic phases, selection for resistance can be extremely strong, with infected individuals producing zero offspring while resistant individuals continue reproducing. This creates selection coefficients approaching 1, the theoretical maximum. But when parasites are absent or inactive, there is no selection on resistance at all. The selective environment oscillates between maximal intensity and complete relaxation. Other systems show more continuous selection. In human immunodeficiency virus, the parasite continually evolves within each infected individual throughout the course of infection. The immune system exerts strong selection on viral populations, favoring viral variants that can escape recognition. The virus maintains extensive antigenic diversity through mechanisms like hypermutation and recombination. Some parasites store large repertoires of antigenic variants. Plasmodium falciparum carries approximately 60 variants of surface proteins, while Trypanosoma brucei maintains several hundred variant surface glycoprotein genes. This escape variant generation, in turn, exerts selection on the immune system, which responds by producing new antibodies and T cells targeting the escaped variants. This intrahost coevolution proceeds without seasonal breaks, creating sustained reciprocal selection for as long as the infection persists. The rate of mutation varies tremendously across host parasite systems and profoundly affects coevolutionary dynamics. RNA viruses like influenza and HIV have extremely high mutation rates, generating new genetic variants constantly. This allows rapid evolution but also imposes a cost, as many mutations reduce viral fitness. Bacteria have intermediate mutation rates, while multicellular hosts typically have much lower mutation rates. These asymmetries in mutation supply mean that in many systems, parasites can evolve new infectivity mechanisms faster than hosts can evolve new resistance mechanisms, creating the temporal lag characteristic of Red Queen dynamics. Experimental evolution studies have revealed how quickly these reciprocal adaptations can accumulate. In one long-term study, researchers allowed bacteria and their phages to co-evolve in the laboratory for over 1,000 bacterial generations. They periodically froze samples of both populations, creating a frozen fossil record of evolutionary change. By reviving these frozen ancestors and testing them against evolved descendants, researchers could measure the tempo of adaptation. The results showed that both bacteria and phages evolved most rapidly during the early generations of coevolution. Bacteria initially evolved resistance to ancestral phages, and phages evolved the ability to infect resistant bacteria. But rather than continuing to escalate indefinitely, the rate of evolutionary change eventually slowed. Both populations reached states where further evolution occurred, but at a reduced pace. This deceleration suggested that the populations were exploring an increasingly constrained space of possible adaptations, with fewer new mutations available that could provide large fitness gains. Importantly, the evolved bacteria didn't show universal resistance to all phages, nor did evolved phages show universal infectivity against all bacteria. Instead, both populations diversified. Multiple bacterial lineages coexisted, each with different resistance profiles. Multiple phage lineages coexisted, each specialized on different bacterial types. This within-population diversity emerged from the coevolutionary process itself, as negative frequency-dependent selection favored rare variants on both sides. The spatial structure of populations adds yet another layer of complexity to these dynamics. In the wild, hosts and parasites don't exist as single panmictic populations. Instead, they're structured into networks of local populations connected by limited migration. A parasite genotype might be common and successful in one location, but rare or absent in another. Host genotypes similarly vary in frequency across space. This geographic structure creates what is known as a geographic mosaic of coevolution. The mosaic has three key elements: selection mosaics, where the strength and direction of selection vary across locations; coevolutionary hotspots and cold spots, where reciprocal selection is intense in some populations but weak or absent in others; and trait remixing through migration, which spreads locally adapted variants across the landscape. In hotspots where parasites are consistently present, coevolution proceeds intensely and hosts evolve higher resistance. In cold spots where parasites frequently go extinct locally, selection for resistance is relaxed or absent, and host populations may lose resistance alleles through drift. Migration between populations can either reinforce or disrupt local adaptation. If migration rates are too high, they prevent local adaptation by constantly introducing maladapted genotypes from other populations. If migration rates are too low, populations may become so genetically isolated that they lose variation needed to respond to selection. Intermediate migration rates can actually enhance coevolutionary dynamics by occasionally introducing novel genetic variants while still allowing local adaptation to proceed. The complexity of these dynamics underscores a crucial point. There is no single pattern of host-parasite coevolution. The Red Queen framework provides a general explanation for why coevolution is ongoing, but the specific patterns depend on the genetic architecture of resistance and infectivity, the strength and constancy of selection, mutation rates, population structure, and numerous other factors. Each host-parasite system represents a unique evolutionary experiment, exploring its own path through the fitness landscape shaped by reciprocal antagonism. What unites all these systems is the reciprocal nature of the evolutionary process. Each adaptation by the host creates selection for counter-adaptation in the parasite. Each counter-adaptation creates selection for further host evolution. This feedback loop ensures that neither party reaches a permanent solution. The arms race may sometimes slow, but it never truly ends. The fluctuating selection pressures, rapid evolutionary responses, and reciprocal genetic changes continue as long as the interaction persists, creating the perpetual motion that defines the Red Queen's domain. Chapter 5. The Diversity Paradox. A single population of Daphnia in a Swiss lake harbors over 20 distinct resistance genotypes. Human populations carry hundreds of different alleles at immune genes. Plant populations separated by mere kilometers show dramatically different arrays of disease resistance variants. This abundance of genetic diversity presents a paradox. If natural selection consistently favors the fittest variants, why doesn't it eliminate this variation? Why do populations retain so many different genetic solutions to the same problem? For most traits, the answer remains elusive or involves multiple contributing factors. But for traits involved in parasite resistance, we now have compelling evidence that parasites themselves actively maintain the genetic diversity they select upon. This isn't a side effect or an incidental consequence. Rather, the maintenance of diversity emerges directly from the evolutionary logic of host-parasite interactions. Parasites create a selective regime where being rare is intrinsically advantageous, and this advantage prevents any single resistant genotype from rising to fixation and dominating the population. The mechanism operates through what ecologists call negative frequency-dependent selection. In host parasite systems, the selective value of a resistance genotype changes constantly depending on its frequency in the population. When a particular resistance genotype is common, parasites experience strong selection to overcome it. Parasites that can infect this common host type have access to abundant hosts, allowing them to reproduce prolifically. As these parasites increase in frequency, the formerly advantageous resistance genotype becomes a liability. Individuals carrying it now face the heaviest parasite burden precisely because they carry the resistance type that parasites have evolved to defeat. But something remarkable happens to rare resistance genotypes during this process. Because they're uncommon in the population, parasites haven't adapted to them. These rare hosts escape infection not because their resistance is intrinsically stronger, but because the parasite population hasn't yet evolved mechanisms to overcome their specific defenses. Being rare confers an advantage. As the common genotype declines under heavy parasitism, rare genotypes increase simply by avoiding this burden. Eventually, what was rare becomes common, and the cycle begins anew. This dynamic creates something fundamentally different from standard directional selection. In directional selection, a beneficial allele rises to fixation, and diversity is lost. In negative frequency-dependent selection, no allele can remain beneficial indefinitely. The very process of increasing in frequency transforms an advantage into a disadvantage. This prevents fixation and instead maintains multiple alleles in stable or cycling frequencies. The population retains polymorphism not despite selection, but because of it. As seen in the New Zealand snail populations and Lake Agelsea Daphnia described in Chapter 2, common clones consistently suffer higher infection rates than rare clones, creating oscillating dynamics where frequencies rise and fall in predictable patterns driven by the lag between host frequency changes and parasite adaptation. The scale of diversity that this process can maintain is remarkable. In some Daphnia populations, over 20 distinct resistance genotypes coexist in a single lake. Each genotype shows different susceptibility profiles to different parasite isolates. Some are resistant to parasites collected early in the season, but susceptible to late-season parasites. Others show the opposite pattern. Still others are resistant to parasites from their own lake, but susceptible to parasites from nearby lakes. This combinatorial diversity, with each genotype occupying a unique niche in resistance space, is exactly what negative frequency-dependent selection predicts. This cycling has profound implications for understanding why sexual reproduction persists. Sexual reproduction doesn't create new resistance alleles from scratch. The alleles already exist in the population, maintained there by frequency-dependent selection. What sex does is recombine these alleles into new multi-locus genotypes. This recombination is crucial because resistance often depends on epistatic interactions among multiple genes. Detailed studies of Daphnia resistance to bacterial parasites revealed that resistance depends on interactions among multiple genes. Specific combinations of alleles at different loci are required to produce the resistant phenotype. This genetic complexity has crucial consequences. When two resistant parents reproduce sexually, the genetic shuffling that occurs during meiosis breaks apart the specific combination of alleles that made the parents resistant. If resistance requires having allele A1 at gene A, allele B2 at gene B, and allele C1 at gene C, then offspring might inherit A1, B2, C2, or A2, B2, C1, or various other combinations. Most of these recombinant genotypes will be less resistant than their parents. This seems paradoxical. Why would breaking apart successful genotypes be advantageous? The answer lies in the coevolutionary dynamics. During a parasite epidemic, selection strongly favors the multi-locus genotype that provides best resistance. Asexual reproduction would faithfully transmit this winning combination, allowing it to dominate. But this creates the perfect target for parasites. Given enough time, parasites will evolve to overcome this single genotype. When that happens, the entire asexual population becomes vulnerable. Sexual reproduction prevents this by continuously regenerating variation. Yes, most recombinant offspring are less fit than their parents in the current selective environment. But some recombinants possess novel multi-locus combinations that parasites haven't encountered. When parasite selection shifts, as it inevitably does when parasites adapt to common host genotypes, these previously unfavored recombinants may suddenly become the most resistant types. Sexual recombination thus restores the variation that selection had depleted, maintaining a reservoir of genetic diversity that allows populations to respond to changing parasite pressure. Experimental evidence supports this mechanism. When researchers allowed Daphnia populations to reproduce sexually after experiencing parasite epidemics, they found that sexually produced offspring were twice as resistant to parasites from the following year compared to asexually produced offspring from the same mothers. This twofold advantage emerged because sexual recombination generated genetic diversity that parasites hadn't yet adapted to defeat. The asexual offspring, genetically identical to their mothers and to each other, presented parasites with a familiar, easily exploited target. The maintenance of diversity through parasite-mediated selection finds its most famous example in human populations. The sickle cell allele in the hemoglobin gene provides protection against malaria at a terrible cost. Individuals carrying 2 copies of the sickle allele suffer severe anemia that often proves fatal without medical intervention. Yet, in regions where malaria is endemic, the sickle allele persists at frequencies of 10 to 20%. This polymorphism exists because individuals carrying 1 copy of the sickle allele and 1 normal copy enjoy both protection against malaria and relatively mild anemia. The heterozygote advantage maintains both alleles in the population despite the severe costs of homozygosity. This represents a special case of balancing selection called overdominance, where the heterozygote has higher fitness than either homozygote. But balancing selection can also arise from frequency dependence without requiring heterozygote advantage. In the major histocompatibility complex genes of vertebrates, hundreds of distinct alleles coexist in many populations. These genes encode proteins that present fragments of pathogens to the immune system, allowing recognition and attack. Different alleles bind and present different pathogen fragments, meaning that individuals with different MHC alleles can recognize and respond to different sets of parasites. The MHC genes show telltale signatures of balancing selection. They exhibit far more variation than neutral genes do. They maintain the same allelic lineages across multiple species through a phenomenon called trans-species polymorphism, with some human MHC alleles being more similar to chimpanzee alleles than to other human alleles. This indicates that the variation is ancient, predating the speciation events that separated these lineages millions of years ago. Only strong balancing selection could maintain specific alleles for such extended periods. Parasites drive this balancing selection through multiple mechanisms. Rare MHC alleles may be favored because they allow recognition of parasites that common alleles miss. Parasites may evolve to avoid presentation by common MHC alleles but remain vulnerable to rare alleles. Additionally, mate choice based on MHC genotype ensures that offspring receive diverse immune capabilities. Individuals can detect MHC genotype through odor and scent, and they preferentially mate with individuals who carry different MHC alleles than themselves. This preference, detectable in both humans and other vertebrates, directly promotes the maintenance of polymorphism by favoring the production of offspring with broader immune recognition capabilities. The geographic distribution of diversity adds another dimension to this picture. In the plant pathogen system of ribwort plantain and powdery mildew studied across Finland, different populations harbor different sets of resistance alleles. A resistance allele common in one population might be rare or absent in neighboring populations just a few kilometers away. This spatial mosaic arose because different pathogen strains dominated in different locations, creating different selective pressures. Where one pathogen strain was common, specific resistance genes were favored. Where a different pathogen strain dominated, different resistance genes provided better protection. This spatial heterogeneity in selection maintains diversity at the species level, even when local populations show reduced diversity. A resistance allele that is rare and declining in one population might be common and favored in another. Migration between populations redistributes this variation, preventing local loss and ensuring that the species as a whole retains a broad portfolio of resistance variants. The species doesn't need to generate new resistance mutations constantly. Instead, it maintains a reservoir of variants that shift in frequency and distribution across space and time. The maintenance of diversity through parasitism contrasts sharply with other evolutionary forces acting on populations. Genetic drift erodes diversity, randomly eliminating alleles, especially from small populations. Directional selection eliminates diversity by favoring one variant and driving alternatives to extinction. Mutation creates diversity, but too slowly to balance these diversity-reducing forces in most cases. Only selection that actively favors multiple alleles can maintain high diversity over long periods. And parasite-mediated frequency-dependent selection provides exactly this. The implications extend to understanding population persistence and adaptation. Populations with high genetic diversity in resistance genes can respond more flexibly to novel parasites. When a new pathogen emerges, some fraction of the population will likely possess resistance alleles that happen to provide some protection, even though those alleles weren't selected for resistance to this specific pathogen. The standing genetic variation maintained by past parasites becomes raw material for adaptation to future parasites. This occurred when West Nile virus first invaded North American bird populations. Populations with greater genetic diversity at immune loci showed better survival and faster evolutionary response to the novel pathogen than populations with reduced diversity. The long-term preservation of polymorphism also has consequences for the evolution of sex itself. If sexual reproduction is maintained primarily by its benefit in combating parasites, then the genetic diversity that parasites maintain is precisely the variation that sex acts upon. A population with no genetic diversity and resistance would gain no benefit from sexual reproduction, as there would be no variation to recombine. The fact that parasites maintain high diversity creates the conditions under which sex remains advantageous. The maintenance of diversity and the maintenance of sex form a reinforcing feedback loop, each supporting the other. Even asexual organisms can maintain diversity under parasite pressure, as shown by bacterial phage experiments described in Chapter 4. However, sexual reproduction changes how that diversity is organized. In asexual populations, diversity exists between lineages. Each clone maintains its own combination of resistance alleles, and this combination is passed intact to all descendants. In sexual populations, diversity exists within the gene pool. The alleles are shuffled each generation, creating new combinations. Sexual populations can therefore explore a much larger space of possible genotypes, accessing combinations that would require multiple specific mutations to arise simultaneously in an asexual population. The preservation of polymorphism through parasite-mediated selection represents one of evolution's most elegant solutions to the problem of maintaining adaptability. Rather than discarding genetic variants that aren't currently optimal, populations retain them as insurance against environmental change. The parasites provide the selection that keeps these variants circulating, preventing their loss to drift while preventing any single variant from fixing. The result is a dynamic equilibrium where diversity persists not despite selection, but because of it, maintained by the very forces that might have been expected to eliminate it. This preserved variation becomes the foundation for continued evolution, ensuring that populations retain the capacity to respond to whatever challenges their parasites present next. Sexual reproduction, despite its enormous costs, persists because parasites make it essential. By constantly shuffling genetic combinations, sex transforms hosts into moving targets that parasites struggle to track. The experimental evidence is compelling. Sexually produced offspring show twice the resistance to co-evolved parasites compared to their asexual siblings, a benefit large enough to overcome the twofold demographic cost of producing males. This isn't evolution towards some distant optimum. It's a perpetual race where standing still means extinction. Parasites maintain the very genetic diversity they select against, creating a dynamic equilibrium where no single solution remains optimal for long. The implications extend beyond explaining sex itself. They reveal that most evolution may not be adaptation to physical environments, but rather endless adjustment to other evolving species. In the Red Queen's realm, survival requires not finding the right answer, but continuously generating new questions. Thank you for listening.